Okay, so our last topic of eukaryotic gene expression are these things called non-coding RNAs, or you might also see them called microRNAs. These were recently discovered, and when I say recent, it probably is in the last 10 to 15 years. And before that, scientists knew that only a small portion of our genome actually is protein coding, so coding for mRNA, and then another small portion codes for the ribosomal RNA and the tRNA. There was a significant amount of the genome that scientists didn't know what it was, so they kind of naively called it junk DNA. But within the last 10, 15 years, scientists have found out that this quote unquote junk DNA actually codes for these really, really, really important non coding RNAs called microRNAs. So when we say non coding, that word just means that they're not responsible for gene expression. They're not responsible for making a protein. Because if we compare them to mRNA, rRNA, and tRNA, all three of those work together to transcribe and translate a gene. So they code for a protein. But these non-coding RNAs, these microRNAs, don't help in transcription and translation at all. In fact, they are actually a regulatory mechanism that can stop gene expression. It can stop transcription and translation. So we call these, as we said, microRNAs. You'll see that shortened to miRNAs, and they can actually even be shortened even further. I've seen people just call them MIRS, MIRS. These are tiny. Okay, so they're shorter in sequence than mRNA, they're shorter in sequence than tRNAs. And when you get to college, if you are taking a genetics course or a molecular biology course, you'll probably be responsible for looking at and knowing how these microRNAs are created, but that really is beyond what we need to know. So all of this up here is how an mRNA is made. This all stuff up here you don't need, okay? But if you're curious, take a look at it. It's a pretty cool mechanism. But what a microRNA is this orange sequence of RNA. So it's still going to be made of A's and U's and C's and G's, and it associates with a protein, and this system right here called the miRNA protein complex acts like a blocker of gene expression. And it can block gene expression in two ways. Way one is this would be floating around the cytoplasm, and if it has a complementary base pair to, I should say complementary base pairing, so all of the pairs line up to the mRNA, it will attach to the mRNA, and that triggers a series of events in the cell where nucleosomes and nucleases come and break down the mRNA, so it's degraded. So when this attaches to an mRNA, it stops transcription or translation because, well, we don't have mRNA anymore. It's gone, right? It's broken down. So that's one area where these microRNAs come into play. And this is very controlled. So when the cell wants to shut this pathway off, when it wants to shut off translation of this mRNA, it will make a microRNA that is exactly complementary to the mRNA. So this is very controlled. It's not random. It's almost like this comes out into the cytoplasm searching for this mRNA to stop translation of it. So that's one area that the microRNAs can play a role. The other area is on the other side. So another way that this can stop gene expression, if you notice that this is mostly complementary base paired, this protein complex, when it attaches to the mRNA, a ribosome is not able to move down the mRNA to translate it. So we call this translation blocking. This is another way that the microRNAs can regulate gene expression. Again, this isn't random. This isn't just something uncontrolled by the cell. All of this is very planned and very controlled and very regulated. So microRNAs, their job is to find mRNAs and stop translation. They can do that either by triggering mRNA to be degraded, or simply they're just blocking the ribosome from doing translation so that mRNA will not be expressed. That really is all we're going to look at for that as far as like exam questions. Now, I do want to take it one step further because this has a very special place in my heart. About 10 years ago, I worked in a genetics lab at Pitt, and we were dealing with and starting to work with these microRNAs as treatment options. This, as a use for humans or other organisms, we call this RNA interference. And instead of testing this on humans, we were testing this on little C. elegans, they're little worms, and the worms that we were working with glowed in the dark. So they were treated with that GFP protein. They were expressing that same protein that your bacteria were expressing. So they were genetically modified to do that. We created microRNAs, fed them, yes, we fed them, to the worms, and those microRNAs went into the worms and blocked the expression of that GFP protein. So once we watched them for a few days, we saw that their glowing stopped. So we call that gene knockdown. We didn't do anything to the DNA, we just blocked translation of that particular gene, and our worms never glowed again because we were blocking them and the expression of that GFP gene. If and when we come back together, I can show you that poster. I think I showed you guys that poster before when we looked at poster making in science. That was one of the things that we did. Another thing that we did with the C. elegans was we worked with a muscle protein. And as you can see in this GIF, that they kind of swim like a snake. They move like a snake. They're very smooth. And one of the other genes that we were knocking down was a muscle protein. So we created a microRNA that was complementary to that mRNA. And when we fed that miRNA to the worms, it blocked the translation of that muscle protein. And unfortunately, the worms didn't move as smoothly as that. They were very jerky, and we could tell that we had affected their muscle proteins because they were no longer moving like this. So we knocked down the expression of that gene. Now, where is this beneficial? Let's say as a human, if we would ever take this to human trials, that you have a defective gene. Maybe you have the cystic fibrosis gene. We could give you microRNAs to slow down or knock down the expression of that gene. All right, do we see where we could kind of apply this? So this is a cool area of genetic research right now that if you're interested in lab work or things like that research, this is a very hot area in science right now. That's pretty cool. So again, as we go back, just so you know what we're talking about here, just kind of know in general what a microRNA is. It's a microRNA. It's an RNA made from a sequence of DNA that we used to call junk. And the function of that microRNA is to stop gene expression with the translation step. It can either do that by tagging the mRNA for breakdown, or it can block translation altogether. So that's really what I want you to know for that. What does a microRNA do? It's another place that we can control our gene expression. All right, today at 2 o'clock, if you're watching this on Thursday, we will be having a Zoom meeting. So if any of this is confusing, come meet with us, come chat with us, and I will be happy to help. See you soon.